Today I will be showing you my 3D model of the KRAS protein. So the KRAS protein is typically located within the cytoplasm, a little more specifically anchored within the inner leaflet of the plasma membrane. The KRAS protein is often associated with lung cancer as it causes rapid mutation of abnormal cells in the lungs, which eventually develops into tumors. And I will be giving you an overview of both the wild type of the protein and the mutation, which I was assigned the G12C mutation, and we're gonna see what it's all about. All right, here is a much up close and personal view of my project. It is made out of pipe cleaner, glue, and a whole lot of flour foam. As we look at the shape, we notice that it is a globular protein, given the fact that it's in the shape of a sphere. The 3D structure of the protein is a tertiary structure. It also has 5 alpha helices, which are in blue, and 6 beta sheets, which are brown. The peptide backbone is also in yellow. And as we look around, we'll note that both switch 1, 2, and the P-loop region are labeled. And these will be important in describing mutations a little bit later in the video. And we'll also note that the protein doesn't have any subunits. And I will be giving you a little bit of a rundown on what the regular functioning is of the KRAS protein. So typically with the wild type, it transmits extracellular signals into intracellular pathways. So it is a part of GTPase, in which it acts as a molecular switch. And when you turn this switch on, it regulates a lot of cell growth and movement within the body. And when it's on, it also utilizes a lot of GTP in order to provide these functions. And it is also, given that it's kind of like a switch, it is also able to be turned off. And it usually uses GDP for this process. So in the active state, it tells the cell to grow and to continue reproducing. So while it's off, it stops sending those signals so that the cell can regulate itself. So the KRAS protein also tends to have a lot of impacts on the organism, including a lot of tissue regeneration, in which it regulates how stem cells renew themselves, which are really important when it comes to wound healing. It also plays an important role in the cardiovascular system because it regulates a lot of blood vessel formation, and it plays a huge role in the maintenance that goes into that. There are various amino acids that are extremely important to the normal amino acid characterization of the KRAS protein, and this includes a couple of nonpolar and polar amino acids. For nonpolar, it includes glycine, leucine, and valine. And glycine is typically very important because it plays a role in allowing to make changes that allows the KRAS protein to adequately perform GTP binding and hydrolysis. Leucine tends to be really hydrophobic, which adds to the characteristic of the structure. Valine is hydrophobic as well as being nonpolar, and it helps stabilize the structure. And another really important one to note is serine, and while this amino acid is polar, it does help it form hydrogen bonds within the structure. Alright, so for this portion of my project, I will be giving you a demonstration of what the G12C protein mutation in action looks like, which I'm very excited to do because I did a lot of research on this in the part 1 portion of this project. Alright, so while we're looking at this, I hope you remember the P-loop region that I did label because it will be important for this demonstration. So at this P-loop region here, this is where the mutation occurs, in which the replacement of glycine at position 12, which I will label here for you, so that it can be a little more easy to visualize. So this glycine that's typically here ends up mutating, and it ends up turning into a cysteine. And we know from earlier that glycine does a really good job of giving the structure flexibility and providing it with what it needs. But if we change it to a cysteine, it causes it to become really overreactive. And this makes the switch remain on without having a method to turn off. And so more cells are being created at a larger rate. And it doesn't really need receptor activation to do it, it just turns on the cellular process. It tends to downregulate other proteins that try to come in and try to control this growth. And typically, in terms of how it impacts the organism, for example, humans, in this G12C mutation, we realize that it begins to form tumors due to the uncontrolled cell growth that's going on. And these tumors intrude into the function and development of a lot of bodily organs that contribute to lung cancer and a lot of other different forms of cancers. So without the given flexibility of the glycine, which provides a lot of flexibility for this molecular switch we have going on, GTPase begins to struggle between being able to be active, which is typically GTP bound, and inactive, which is usually a GDP bound, which is not very good for the hydrolysis process because it slows down. Or even in some cases it becomes inhibited, which creates this like active, like this on switch that's permanent. And while we have cysteine here, it's also a good thing to note that now that we have this mutated amino acid, it brings with it its new characteristics. It's large and it's polar, and it also introduces a new thiol group. And the sulfur atom within it is reactive, and it allows the structure to now form disulfide bonds and have more interactions that it wasn't able to have before. I really hope that you were able to take a really good moment and review with me and go over the protein structure and how it impacts a lot of its function. But now that all of that's said and done, I do want to go over a few of the limitations my model did have and that I did realize while I was making. I do realize that the backbone structure of this model is a little inaccurate, given that a lot of the backbone down here is not very visible, and it's very difficult to gauge in video both and in person, which can actually stray it a little bit from its original model due to the limited amount of space given by the flower foam. I will also say that it's also really difficult to imagine the side chains in this. All it really shows is a couple of the helices and a couple of other structures, but it doesn't really get into the nitty-gritty that I think I would have preferred when it comes to explaining this to someone. And you also, this is more of a personal thing, but I also feel like I didn't really have enough space, both because the creation is a little bit fragile, to incorporate GDP and GTP molecules so that you could visualize it a little bit better, be able to create this molecular switch, but out of fear that this was not going to remain together and trying to demonstrate that to you, I feel like it lacked that true real process in action.